Right, so I think we'll start now. So it's a great pleasure to reintroduce Saurabh. You all heard him talk about the basics of probability statistics and machine learning last week, and also an enjoyable after dinner session for us, mid-morning session for him. And now he's back to give us a research seminar on things he's been doing recently. So over to you, Saurabh.
Thanks, Rahul. Thanks to you and the other organizers for inviting me at this amazing event. I plan to talk today about some ongoing work in my lab in the area of single-cell omics, where we use a variety of statistical and machine learning tools to glean new insights about molecular and cellular biology. So let's start with a quick look at one genre of new technologies revolutionizing biology today. So the revolution really started about 10 years ago with emerging technologies for large-scale single-cell biology. One technology after another kept emerging, giving us views of the contents of individual cells in increasingly greater levels of detail. The most popular among these technologies, single-cell RNA-seq, gave us expression levels, that is numbers of transcripts of every gene in individual cells for thousands of cells. Another name for this technology is single-cell transcriptomics, that is measurement of the transcriptome at single-cell resolution. The next big milestone was in the form of spatial single-cell transcriptomics, which provided us, in addition to the transcriptome of each cell, its spatial location as well, in the form of XY coordinates in the tissue. In fact, this technology was declared the method of the year 2 or 3 years ago by the Nature journal. Now, believe it or not, even that was not the limit of advances, and what came next, quite mind-bogglingly, was spatial transcriptomics with single molecule resolution. To explain with an example, this is a piece of the data provided by this kind of technology, one of which is called MERFISH. And what you're seeing are views inside cancer cells. Each dot here is a transcript of a gene. The right panel shows a zoomed in version of the little square on the left. And the color of each dot shows the identity of the gene whose copy it is. Now, such data can be easily represented in a data frame or spreadsheet, with each row representing a single transcript, the gene it is a copy of, the cell it is located in, and the spatial coordinates of that particular transcript. So now you've seen glimpses of the magical new data types, which are being collected in numerous different biological contexts. And it seems that these technologies are ahead of us analysts who are asking, well, what can we do with these sorts of data? I'll talk about 2 interesting types of biological inquiries that we have pursued in our lab using such high-resolution, large-scale data sets. First, can we find patterns in which genes are where, and with which other genes do they hang out? And second, what are the control mechanisms underlying the high and low expression levels of different genes? These 2 inquiries have been of interest to biologists for decades now. The first class of questions about who is where and with whom goes by formal names like subcellular localization and RNA interaction, while the latter question, the who's controlling whom, pertains to the biological concept of gene regulatory networks. So we're ready now to dive into the first investigation of cellular ongoings, who's where, with whom. And I want to add that this was and is a journey into a completely unknown territory for us, where we didn't know going in what we were going to find, and the data was our only guide. As I said, whose where, that is RNA localization in a cell, is a process of great interest to biologists. And a variety of mechanisms are known that can cause a gene's transcript to be preferentially localized to certain parts of the cell at a given time. These include processes like nuclear export, which can be regulated, condensate or particle formation, intracellular transport, translation, degradation, etc. So there's a lot going on inside the cell, resulting in non-random positioning or localization of different RNA species, different genes transcripts in different parts of the cell. Similarly, many different biological studies have cataloged the different types of interactions between RNAs and other macromolecules in the cell, from proteins to lipids, and their multifarious functions in normal cellular processes and in pathologies. We were approaching this from a data and analysis point of view, and we decided to focus on RNA-RNA co-localization. And the best way to explain what I mean is with these samples. Each of these is an actual cell in the data with transcripts of two particular genes named PRKDC and FASN, shown in blue and pink respectively. Transcripts of all other genes are shown as gray dots. And you can probably appreciate, just by looking at these without using any sophisticated statistics, that the transcripts of these two genes tend to be around each other. That's what we mean, roughly speaking, by RNA-RNA co-localization. We defined the concept of decolocalization, to try to make this idea more concrete, as the tendency of two genes, rather their corresponding RNAs, to occur within distance d of each other. And that's to avoid ambiguities and semantic conflicts with the term co-localization itself, which already means something else and more precise. Our goal was to find all gene pairs that exhibit such decolocalization. And we believed that this phenomenon could point us to RNA interactions, localization, and function. Now, at the heart of our approach were two newly developed statistical tests. The first one, called the proximal pairs or PP test, detects if a gene pair decolocalizes in a single cell. In this test, we look at transcripts of that gene pair and count how many pairs of transcripts are within distance d. And then we obtain a p-value for that count using a binomial test that is suitably, a binomial distribution that is suitably parameterized. The second test, called the conditional Poisson binomial test, detects persistent co-localization of the same gene pair across many cells. These tests took, and I won't go into the details of this particular test, except to say that it counts the number of cells in which the PP test declares a gene pair to be co-localized, and then obtains a p-value for that count.
Saurabh, sorry to interrupt. Naive question. What is the significance of transcripts co-localizing? Does it imply protein co-localization, or is there? Yeah.
That's a great question, and I will. Much of this talk will try to find out what these. Firstly, to see if we do find statistical co-localization, and then what might those events be pointing to, what biology might be reflected in them. And You could imagine, of course, that RNA-RNA interaction could reflect in such co-localization, but to the extent that RNAs code for proteins, whether protein interaction may reflect in their corresponding RNAs also co-localizing is not a given at all. So it's very much a discovery process, and we are in kind of collecting such evidences.
Can I ask another question?
Yeah, sure.
Thank you, Saurabh. So is this co-localization 3D, or do you see a possibility of false neighbors on 2D?
So that's a great question too. The tests I'm describing are with 2D images. Now, the technology does, if used appropriately, can provide you 3D data with multiple Z planes of the same cells. And we have 3D versions of these tests as well, which I'm not going to go into detail. But I should say that it's not truly 3D. It's 2D planes going through the third dimension.
Thank you.
Okay.
One more question. Do you have microRNAs also in this?
So that's a great question too. No, this technology is actually based on a pre-designed panel of genes. So it doesn't probe all RNAs in an unbiased manner. It's imaging based. And by the technology, I mean Murphish kind of technologies. It's imaging based, and you have to pre-select a set of genes that you want to examine. And typically, you choose genes that have longer RNAs and RNAs that are not too abundant for the technology to work properly. So microRNAs are not represented in those panels.
I see.
But link RNAs can be.
Right.
One more question.
Sure.
How is the cutoff distance D is decided in the proximal pairs test?
Right. So I was not planning to talk about it. There is no golden rule for deciding that D. What we have done when using these tests and these algorithms is to try a few different values of D and assess the false positive rates of discovery at these different distances. The distance threshold will also depend on the cell sizes. Sometimes these images are taken on what's it called? Cells that have been expanded artificially for easier viewing, and so the distance thresholds have to kind of reflect the real estate of the cell within which the co-localization is happening.
So can't it get estimated experimentally?
Well, it depends on what you want to use the co-localized pairs for, right? So if you were only interested in detecting co-localizing gene pairs that co-localization arising from direct RNA-RNA interaction, then you would probably want a very small d, that is at the resolution that captures direct interactions. But if you're interested in other sources of co-localization, then you might be interested in a larger value of this threshold as well. So this is, in that sense, it remains arbitrary, at least for now. We haven't figured out a systematic way to define, to determine the right D.
Okay, thanks.
Okay, so these tests actually took a lot of effort to design and perfect. We borrowed from the rich literature on spatial statistics to the extent possible, but there were many things that were, many challenges that were unique to this sort of molecular data, not faced by the spatial statistics applications. And we had to take care of things like the non-random spatial landscape within each cell, the varying shapes and sizes of cells, and other kinds of heterogeneity across cells. Okay, so it is, of course, useful to know more about a decolocalizing gene pair than a mere p-value. So we also implemented a simple categorization scheme whereby each pair is annotated by the cellular region it is generally found in. So this is, again, a statistical call for whether a gene pair that's significantly colocalizing tends to do so in, let's say, the nucleus or at the nuclear membrane or cytoplasm or the cell membrane. And here's an example of decolocalization in a single cell detected that fell into each of these four categories. And in our analysis, the largest number of colocalizing pairs were observed in the perinuclear and nuclear regions, though there were interesting examples in the other two categories as well. An obvious challenge one faces when embarking on biological discovery, especially in a poorly mapped domain of knowledge, is validation. There simply isn't any benchmark or ground truth here to perform the systematic assessments, unbiased, objective assessments of the predictions made by our methods. So we did the next best thing, which is to use randomizations of the data to assess false positive rates, and found that several hundreds of gene pairs could be designated significant at levels where the estimated false positive rate is less than 5%, and that these false positive rates also depend on the D value used in the statistical tests. And by the way, this randomization refers to taking the entire data set, keeping the transcripts locations and their counts unchanged, simply shuffling the gene labels of the transcripts among each other so that the structure of the data remains preserved and only the identities of transcripts are garbled. And that's how we set a null distribution that allowed us to assess false positive rates. We assessed replicability of these decolocalized gene pairs across different replicates of the data set, which thankfully were available to us, noting a replicability of about 80% across these replicates, compared to about to less than 10% expected by chance. And we even generated our own MERFISH data sets and on U2OS cell lines, and checked for replicability between our data and published data from the Zhuang lab on U2OS cell lines. And the replicability was around 40% compared to about 5% by chance. Interested in understanding, this sort of alludes to the question Rahul asked earlier, understanding why we see decolocalization so commonly, we performed corroborations of these gene pairs with various databases of gene-gene relationships. And among these, for instance, we found that genes annotated with the same function tend to colocalize more often than random expectation. We also found that RNAs that are likely to bind each other, according to well-established RNA-RNA interaction prediction software, also have a significant tendency to colocalize. In other words, decolocalization may indeed be pointing us to functional relationships, either direct interaction or just co-function. An exciting example of a colocalized gene pair that we explored in greater detail is the pair MALAT1 It's a long non-coding RNA or link RNA, and SRRM2, messenger RNA. These are three examples of cells out of hundreds exhibiting their co-localization as detected by the PP test. This was interesting to us because of some background knowledge about these molecules from the literature. So MALAT1 is known to at least partially localize to structures in the nucleus called nuclear speckles. And SRRM2 protein is a core component of nuclear speckles as well. So, of course, we were seeing co-localization at the RNA level, and this observation from the literature was about the SRRM2 protein. And so we hypothesized that MALAT1 and SRRM2 RNA co-localize in nuclear speckles, even though our analysis had no idea about the existence of these speckles. And indeed, carefully designed experiments that co-stained these molecules with a protein marker of nuclear speckles that's called SON, confirmed our hypothesis. And I find this to be cool because it's an example of how we can use who's with whom and answers to that question to discover something about who's where, in this case, molecules that are localizing to nuclear speckles. Okay, so our next question was whether these extensive co-localization relationships we were finding are universal or whether they are specific to particular cell types, such as neurons versus astrocytes versus oligodendrocytes, etc., when looking at brain samples. Single-cell transcriptomics data are often used to identify the cell types in a sample and then plot the cells in a low-dimensional projection, such as this one, with colors indicating the cell types. So each dot here is a cell. These are plotted in an abstract two-dimensional space, and in such a way that similar cells will kind of get plotted near each other. And you can then follow it up by recognizing each cluster as being a particular cell type. So this is already done on the data sets that we were working with. We then examined for each co-localized gene pair exactly which cells the pair co-localizes in, and whether those cells tend to be of a particular type. Indeed, this was the case for several gene pairs, such as this particular pair, which co-localizes in the cells marked with black dots in this figure. And you can see that the black dots kind of crowd more in this region of this plot, which is the inhibitory neurons in the data. So in other words, this particular gene pair co-localization is enriched in or specific to inhibitory neurons. On the other hand, there were also gene pairs that co-localize more universally and not preferentially in any particular cell type. I have a note here on the technical challenge associated with this analysis. A co-localizing gene pair may appear to co-localize preferentially in a cell type simply because one or both genes in that pair is preferentially expressed in that cell type. So if a gene is a marker of a cell type, only expressed in that cell type, then if you see a gene pair co-localizing and one of the genes is that marker gene, then it's no surprise if the co-localization is also specific to that cell type. So we did address this confounding effect by a suitable adjustment using a generalized hypergeometric test, which I'm not going to go into more details of. All right, so next we asked whether these subcellular co-localization patterns we were observing in many, many cells had a higher level spatial pattern at the level of the whole tissue. And this could be an exciting finding and not surprising since we were analyzing data from the brain in this analysis, in particular, the preoptic region of the hypothalamus in a mouse brain. And the brain is a fertile ground for spatial phenomena, so to speak. This panel shows all the cells in our field of view. So all the gray dots here are the individual cells in the data set, the particular field of view we were analyzing. Now consider the cells where a particular decolocalized gene pair identified by us actually colocalizes as per the PP test. Those are shown as blue dots on the same field of view. And I guess you can appreciate that this is not a random spatial distribution of blue dots, and that there is a tissue or sample level spatial patterning going on with this decolocalization event. This is the kind of spatial patterning we sought to identify automatically, again, without really knowing for sure if we'll find them. We developed a specialized probabilistic model for this purpose, built around a graph representation of the data, with each node in the graph being a cell and its spatial neighbors being adjacent in the graph. We set the probability of observing a gene pair's colocalization, that is seeing a blue dot here, to be dependent on seeing colocalization in neighboring cells or nearby cells. And so with this probability model defined, we did a maximum likelihood parameter estimation and used a likelihood ratio test to infer significant cases of spatial patterning, tissue level spatial patterning of intracellular colocalization of a gene pair. And indeed, we found several examples of such pairs, such as this one. This is a gene pair that colocalizes in the cells shown in blue dots here. And this particular, you can see that it is a very precise part of the tissue where this colocalization is being observed. And similarly here also, it may not be as precise as this one, but the blue dots are clearly not uniformly distributed overall and tend to be enriched in these regions of the tissue. All right, so, and I think that these ab initio discovered examples of tissue level spatial patterning of colocalization events can be very interesting starting points for follow-up studies. So far, I've been talking solely about pairs of genes or RNAs that colocalize, and it was natural to try to extend the approach beyond the pairwise level and ask if entire modules of genes, sets of genes, colocalize with each other. And a simple way to go about this is to consider the matrix of pairwise colocalization strengths. So the rows and columns of this matrix are genes, it's the same genes, and the color intensity shows the strength of colocalization, decolocalization of each gene pair. And then we can just cluster such a matrix and find these subsets of genes that colocalize with each other. So they will come out as diagonally located blocks in this sort of a matrix. And we found two such modules, which we labeled as M1 and M2 in this particular data set. And as is common in bioinformatics, we tested for functional enrichments in these modules, noting, for instance, that genes of module M1 are enriched for actin and cytoskeletal organization. So that's another way to go about hypothesizing why and where from these colocalization events arise.
Question
and here
again.
are three. Yeah, Rahul?
Yeah, sorry. So two questions really. One is I see some off-diagonal blocks which should indicate one group of genes that co-localizes with a different group of genes, right?
That's right.
Is that an interesting thing to look at?
I don't remember what we did with that. So it could be, let's see. So in this case, here's the off-diagonal block, and this set of genes is co-localizing with this other set of genes. Yeah, we haven't gone deeper into that. Again, as you know, Rahul, in other instances of such matrices being analyzed, people do try to interpret these off-diagonal blocks as well. We just haven't had the time to do that. But
and also,
yeah.
second question, have you looked at transcripts that avoid each other, that localize less often than you might expect?
Right. No, we haven't. And let's see. So I think, yeah, it could be done. My gut reaction is that the PP test that tries to detect these unusual co-localization patterns in a single cell will just not have the power to detect an under-representation of co-localizing pairs of transcripts, because usually the expected numbers of pairs by chance are small enough that to detect a significant departure below that expectation is not going to be possible. You'll hit zero before that happens. So that's my first reaction. In some cases, for some gene pairs, maybe the expectation, a random expectation in the cell is higher, and it may be possible to detect underrepresentation. Does that answer your question?
Yes, I think. Thank you. Do you have?
Okay.
Just
All
one
right.
more question.
So
One more
was there
question.
another question?
Yes.
Yeah.
So the technology is agnostic to whether the RNA is in the nucleus or the cytoplasm?
That is correct. So the imaging technology, the Murfish technology, is agnostic to the nucleus versus cytoplasmic location of transcripts.
So
Yeah.
if you're in the nucleus and you're close to where you're being transcribed, I mean, the RNA will be close to its, location in the chromatin.
Yeah.
So genes that are close to each other on the chromatin, do they seem to be co-localized just because they would be transcribed continuously? Right. And
yeah, that's a great comment. So I can only partially answer with one of our experiences around that issue. So some genes, just statistically, were enriched in the nucleus. For example, the link RNA MALAT1 is known to be enriched in the nucleus, and that can throw the statistics off. So we were approaching this purely from a statistical point of view, and we had to be cautious of all kinds of artifacts in our statistical findings. One of the major artifacts or stumbling blocks is when you have this sort of nuclear enrichment of certain genes that throws off some of the assumptions about randomness made by the statistical tests. And so what we did to address that was to provide a separate mode of analysis in our software tool that will limit the analysis to the nuclear parts only. And that's the best we could do. It's very hard to adjust the statistics for the non-random split between nucleus and cytoplasm, and still look at the whole data. So our analysis has two modes of statistics. One is the whole cell level analysis, and one is nucleus only analysis.
Yeah, makes sense. Thank you.
So talking about modules of decolocalization, in a complementary approach to finding such modules, we represented each cell as a graph with edges representing the colocalizations detected in it. So each cell, the nodes here would be genes, and an edge would be placed if that gene pair colocalizes in that cell. and then performed frequent subgraph mining. This is something that's received a lot of attention in the past in the web mining literature, and we used algorithms from that literature to find frequent subgraphs in these many cellular graphs. That gave us a very specific module of 4 genes for this particular data set, where every pair of genes is co-localized in many cells. And again, we found interesting biological interpretations of this module when examining gene functions. And here's a sample visual of this module with the dots showing the transcripts of these 4 genes in 4 different colors, and just 2 cells being shown here. But there were like about 100 such cells where this sort of modular co-localization was observed. Okay, so this is a summary of what I have described so far. We have developed a suite of tools, a toolkit, that is, which we call INSTANT, that can analyze spatial transcriptomics data of subcellular resolution to discover a large number of decolocalized gene pairs, annotate the cellular regions where they tend to be found, find out if they are cell type specific or spatially patterned at the whole sample level, and also report on modules or sets of many such genes. We have more recently also used INSTANT to analyze subcellular spatial transcriptomics data obtained from other similar technologies, not MERFISH, to demonstrate its wider applicability. And one such technology is called SeqFish or SeqFish Plus. The data set we were looking at was on a cell line of mouse fibroblast origins, and nearly 10,000 genes were probed in this tour de force work. And the instant CPB test revealed more than 100 decolocalized gene pairs at a very strong p-value of 10 to the power minus 10, using a D threshold of 2 microns. And that's what's shown here, all the gene pairs that were significantly co-localized. And the ones marked in green here are those we found to be involved in extracellular matrix function. So it was quite remarkable to us that so many genes involved in extracellular matrix function and regulation had their RNAs co-localizing in this particular analysis. And I'm not going to go deeper into this. Again, I know very little, but my limited literature search suggests that there may be localized protein synthesis going on related to the extracellular matrix function. And that localized protein synthesis may result in the corresponding RNAs being enriched or localized in non-random ways, maybe near the periphery of the cell. And that's what's causing this extensive co-localization. We have also, so the technology that I'm talking about, while in its early days, has already made it to the commercialization stage also, and is available from 10x Genomics as the Xenium technology. And the 10x Genomics folks have made a few data sets of the Xenium technology available freely. And we analyzed one such data set on the mouse brain and identified decolocalized gene pairs that were specific to a particular region of the hippocampus. And we have some anecdotal literature evidence for why, what sort of functions might be causing them to co-localize. In particular, there's evidence of several of these to be proteins that localize in dendrites of the CA3 region, and some of them are known to have local translation as well. And so that might be the reason why the co-localization is observed. Okay, so let's see. I will skip the next part of about 2 or 3 slides on ongoing work, where we're trying to find to do subcellular segmentation using this sort of data. And yeah, I'll skip this. I'll just say that we saw that example of co-localization of MALAT1 and SRRM2, leading us to the discovery that both of them are localized in nuclear speckles, even though the analysis did not know about nuclear speckles. So the motivation for us was whether we can automatically discover subcellular regions or structures by unsupervised learning techniques applied to these sorts of data sets. And so, as I said, I unfortunately have to skip this because I want to go on to the second part of the talk, which is about regulatory networks. Okay, so to summarize this first part, we have developed this toolkit for discovery of gene pairs and modules that co-localize, and made some interesting findings by applying this toolkit to a variety of different subcellular spatial transcriptomics data sets. All right, so in the remaining time, I will talk about this, the second major question I outlined at the beginning, that of finding out who's controlling whom in cells. Now, this is a classic bioinformatics problem. Many machine learning techniques have been proposed for it, and what I'm presenting here will be a fresh take on an old problem that sprinkles some elements of an exciting area called causal inference into the pot. Okay, so I should preface this segment by saying that gene control mechanisms, known as gene regulatory mechanisms, are super important. The biologists among you surely know that, and keep coming up as central to a wide array of biological contexts, from development to disease to behavior. The bioinformatics challenge here is to start with expression data of the sort I introduced earlier today. That is a matrix of gene expression values of each gene in each cell, and infer the so-called gene regulatory network, or GRN, which identifies the regulator genes called transcription factors, or TFs, that regulate any given target gene. Okay, so that's what we want to infer from the expression data. Now, a specific version of this GRN reconstruction problem is that we want to infer the control mechanisms in 2 different biological conditions, say treatment and control, or disease and healthy, and see if there are changes in these control mechanisms between those 2 conditions. This problem is referred to as differential GRN reconstruction, and is what I'm going to talk about next. Was there a question? Okay, so we want to develop a method that takes as input expression data in 2 different conditions and produces differential edges of gene regulation, that such as this TF regulating this gene in condition 1, but not in condition 2. A whole family of approaches have been proposed for this problem that infer the strength of each GRN edge based on correlations between regulators and targets, and devise tests for significant differences between the 2 conditions. So you can, these betas are the strengths of evidence for TFI regulating gene J in each condition separately, and then you can take the difference of those and do statistical tests for whether this difference is large. And that's how differential GRN reconstruction has been approached in the past. An alternative approach that is surprisingly less explored is the following. So you model, you model the gene's expression as a function of the TF's expressions, and then once you've trained such a model, often a machine learning model, you can calculate what's called the feature importance, the importance of each TF as a predictor in the model to get these beta values. And then you can, again, compare those strengths between the two data sets or two conditions to infer changes in regulation. So a little more, let's talk a little more about these feature importance scores. This is in particular because it's an intensely researched topic in machine learning today, and it pertains to what's being called interpretable AI. So in a predictive model that uses a number of variables or features to predict something, how important is any one of those predictor variables? And a popular scheme for feature importance scoring is something called the SHAP score from Lee and Lundberg, inspired by game theoretic ideas and the Shapley value of a player's contribution to a game. I will not go into details of that, except just I hope you can take it at face value that there is this popular score called the SHAP score that assesses the importance of each feature or each predictor variable in a machine learning model. So it is this SHAP score of feature importance that we decided to use in our GRN and ultimately differential GRN reconstruction. And the reason we did so is because we found that the SHAP score solves a precise causal inference problem that pertains to the transcription factor's regulatory influence on a gene. In particular, we first defined a causal estimand or quantity that reflects the strength of a TF's control on a gene using Pearl's do calculus. I'll explain this in a bit. Separately, we trained a machine learning model, in particular a random forest or a neural network, F, to predict the gene's expression from the TF's expression. We used the SHAP score to quantify the contribution of each TF to the model's prediction for a particular sample or cell. And most importantly, we theoretically proved that under certain conditions, this SHAP score estimates the causal estimand we define in the first step. So what is this causal estimand that reflects a TF's influence on a target gene? Sorry for the math here. I just want to say that this was a challenge that forced us to think about what is meant by a TF regulating a gene in a particular context and try to formalize it as precisely as possible mathematically. So this causal estimand, we call it the local treatment effect, or LTE, inspired by the average treatment effect that's used in causal inference language, or even in randomized controlled trials. This quantity quantifies the influence of regulator T on a particular gene Y for a particular cell represented by the X vector. So it is trying to measure a TF's influence on a gene's expression in a single cell. It is defined as the difference of two terms. The first one here being the expected gene expression if you could do a magical intervention on a single cell and set that TF's level to the level that's seen in the cell of interest. That's what this do notation here represents. And the second term is the expected gene expression when a similar intervention is done to set the TF's level to some reference value, such as 0. To me, and I hope to some of you, this is an intuitive definition aligned with how a biologist determines a regulator of a gene through experimental interventions. For instance, this part is where you do a knockout of a transcription factor and examine the gene's expression level and compare it to the expression level in the wild type condition. Except that this definition is in the context of a single cell. And I understand that this is just a thought experiment because you can't really do that sort of knockout on one cell at a time. This causal estimand and its sorry. Okay, I should have. This causal estimand and its estimability using an accurate machine learning model and the SHAP feature importance score is at the heart of our approach called SIMLA. First, we train a machine learning model to predict a gene's expression using all candidate regulators and do so separately for each condition. So the treatment could be C equals 1 and the control could be C equals 0. So we'll get a separate machine learning model F sub C for each condition C. Then we calculate the SHAP score of feature or transcription factor T on each of these for each of these two models. And then we take the difference between the SHAP score cell by cell and use the L2 norm of these differences as the measure of that TF's differential importance or to the gene's expression between the two conditions. Okay, so that's the method, and we needed next to evaluate it. And the standard approach for such evaluations today involves working with synthetic data, which is what we did, and showed that SIMLA exhibits substantially greater accuracy over state-of-the-art co-expression-based methods for differential network inference. And the scatter plots here show the extent of that advantage over the second best method in the comparison. Similar comparisons to limited, the limited few machine learning based models that were available gave us similarly encouraging results about SIMLA's accuracy. And as a demonstration of its utility for real data, we applied SIMLA to characterize differential regulation underlying Alzheimer's disease, analyzing single cell expression data from 22 individuals in each of two groups based on diagnosis at the time of death. We combined SIMLA-derived differential GRN information with published brain GRN for multiple psychiatric disorders as derived by the Psych-ENCODE Consortium. And this figure summarizes the transcription factors identified by this integrative analysis with the sizes of the circles representing the number of genes regulated by each TF and the two axes representing the strength of evidence from SIMLA, that's the differential GRN confidence, and the Psych-ENCODE. This is a closer look at the same results, focusing on three of the key regulators, which were CREB3 and NeuroD6 and ELK1, and listing its target genes that are targeted or not targeted specifically in AD patients. Now, this work is merely a stepping stone in a long journey, and several challenges face us. So, for example, the differential GRN reconstruction I showed was on single cell data from scRNA-seq, but without any spatial aspects. And we'd like to include spatial information at the cellular and subcellular level in GRN reconstruction, and we are working on that problem. And also, as some of you might know, there is a trend towards combining single cell epigenomics data, such as ATAC-seq, with transcriptomics data, and use such multi-omics data sets for GRN inference. Finally, we'd like to combine the directions I outlined in the two halves of the talk, the who's where with whom part with who's controlling whom, to get a holistic picture. Okay, so there's no question. So this is my last slide. There's no question today about the incredible power of machine learning and statistics for facilitating biological discovery. Many people have a lot of thoughts about this, and I have a couple, perhaps obvious thoughts as well. There is an increasing demand for unsupervised learning methods that can detect patterns from data without being given examples of that patterns to begin with. So, for instance, the first half of the talk was, in some sense, an unsupervised learning approach. We were trying to find these co-localization patterns But it's not like we were given several examples of gene pairs that co-localize and trained a model on that. No, we were trying to just discover those kinds of patterns ab initio. And so there is an increasing demand of such methods, especially in biology and in molecular biology, but we need to avoid getting overboard. And there's some nice discussion of how you can easily get into slippery slopes when doing unsupervised learning. Also, I'd like to do a shout out for the field of causal inference and say that it has several ideas that machine learning people working on biology can benefit from. After all, we would like to go beyond mere correlations to actual causal relationships between molecules. And so that's where this sort of causal inference ideas can come in. Also, multi-omics views can help prune spurious relationships that are detected a dime a dozen in machine learning-based discovery efforts. Also, we'd like to support constraining machine learning models with physics-based ideas as well. That in some cases has already proven to be an effective strategy. And I'd like to thank the people who've done the work that I presented. So Instant was done by a group of graduate students and 2 PIs collaborating with me. Simla was done by Payam Dibania, and subcellular segmentation was done by Abhishek Ojha. And I'd also like to thank my employers and the funders. Thanks very much.
Thank you very much, Saurabh. And we have time for a few questions. Yeah.
Thanks, Rahul. Thank you, Saurabh. Actually, I have a question from the first part of the talk and a couple from the second. I'll begin with the second part first. Okay. So when you I did not quite understand the model formulation in the causal inference part. So was it done as a linear model when you did the do calculus, including all the transcription factors? Or was there some sophisticated approach where, for example,
right? So yeah, I again apologize. I wasn't expecting to get the details across in this condensed talk, but the paper is on archive if you want to take a look at it. And it's, if you just search for Simla and my name, that should take you there. So the machine learning model that we train here to predict a gene's expression from TF expression levels, we played with and tried ultimately used a simple multi-layer perceptron and alternatively a random forest. So basically any ML model for which the SHAP score implementation exists can be used here. And the tool actually is implemented in a modular way so that you can replace the ML model with another as long as there is SHAP implementation.
Thanks, Saurabh. But do you? But would there be a possibility of confounding in such models? And have you considered directed acyclic graphs or Bayesian networks to do that in the more? Yeah.
Right. So there were very specific assumptions that we had to make in claiming that the LTE that we were defining is in fact a causal quantity. And one of those was the absence of cycles in the underlying graph. Also, the whole method works if the ML model is accurately trained. And so if the ML model is misspecified or mistrained, then that can also break down the claims made by our work.
Thank you. And as a follow-up to the LTE question, so in clinical medicine, we usually now move from ATEs to ATETs, the average treatment effect on the treated. Is there something analogous in single cell where you can actually look at wherever the transcription factor was active, what was the effect on that kind? Is it even possible to do that in your settings or in general in these kind of data sets?
Right. So I didn't know about the ATET. I will look into it. But what we did was, so the two terms here, the first term says, what's the gene expression if you intervene and set the transcription factor at the level it is seen in that cell? This sounds kind of specious because, after all, it is, why do you need to intervene? But this is just saying that you force the TF to be at that level that it's seen at, and force the other TFs to be at the levels they are seen at. So this is like the cell as observed. And then you compare it to an intervention where you set the TF to 0 or some other reference level, while also forcing the other TFs to remain unchanged. So this contrast, the only difference in the right side is that the TF of interest has been zeroed out or set to a reference value in the second term, while everything else is unchanged. And so maybe the first term is kind of like what, I don't know enough about the ATET to say if it's similar or not, but this is how we define. And it was also, this is the definition that will allow us to cast the SHAP score, which is, recall that is something that's coming from the game theory literature. The SHAP score can be precisely claimed to be this LTE. And that was the cool new finding of our work. And that works only with this definition.
Got it. I'll read the paper and come back if I have some further questions. My last question is about the first part of the talk, and apologies if I've missed it. So when you were looking at decolocalization and looking at the regulatory networks in Xenon data that you were showing as well, as in standard enrichment analysis, we have a negative background set to compare against and hypergeometric distributions to sample from. Is there something similar that you did to show that what was the probability of co-localization in a random negative background set while computing these statistics?
Right. So, by the way, is my
no?
Am I still sharing?
No, the screen is.
Oh, sorry about that. Sorry. Okay, so this is the statistical test. That is this what you're talking about? How does this assess the p-value of co-localization?
Right. So yeah, this would be helpful if you could maybe. Apologies if I.
Right. No, absolutely. So what we do is we look at all the transcripts in this one cell. Can you tell me what you are able to see? Can you see my mouse, my cursor?
Yes.
Okay. So there's one cell and there's a whole bunch of dots in various colors. You ignore the colors, you take all transcripts in that cell, look at all pairs distances, and build this null distribution of that cell's inter-transcript distances. Okay, and then you see what fraction of all pairs falls within distance D. Okay, these are transcript pairs. And so you now have a sense of what's the random chance of any two transcript pairs chosen at random to be within distance D in this particular cell, given how its transcripts are distributed. That fraction becomes the heads probability in my binomial test. So if in this cell I see two pairs of transcripts that are for my red gene and blue gene that are within distance D, and there happens to be 3 transcripts of the red gene and 2 transcripts of the blue gene, that's 6 pairs in all, of which 2 are within distance D, then I can do a simple binomial test with 6-2 and this probability that we inferred from the background distribution. So that's the way we use the cell's known transcript distribution in space to define the null distribution.
Right. And perhaps my question was on the lines of this null distribution would also correspond to the enrichment kind of the analysis that you were showing, that there may be some processes that may be enriched. So this is a statistical test to see what's the p-value for any two transcripts coming together. But do you have some similar?
I think in the interest of time, I would say take it offline, but it has to be online, but you can take it online somewhere else. Yeah. Any further questions? Yeah, it's an interesting topic on how to get the null set, and maybe I would also join in, but okay. So let's thank Saurabh again. Thank you very much for joining us.
Thanks very much for your
at some unearthly hour at your time. So we'll break for coffee and meet at 11.